9th and 10th grade channel. I'm your biology teacher Aishwarya.
And hi everyone, I'm your teacher Ankita and we welcome you in today's class.
So I hope all of you today here are excited because today we are going to be doing some important experiments which are there from NCERT. And of course, along with this, on most demand, we are also going to be discussing some of the important diagrams as well. What you will require to learn for drawing and what you will be needing for labeling, and of course, ma'am, what can come as diagram based questions, right? So I hope all of you here are excited and I hope all of you can see us, you can hear us, and our screen is visible. If it is, everybody give us a quick thumbs up in the chat.
Yes everyone, so please make sure to tell us whether you can see us and can hear us. We can see your messages everyone. Good evening, welcome to the class. Hello everyone, it will be difficult for us to call out each of your name. So please, sorry from our side, but we can see your messages. So if you have any doubts, you can definitely write here and we'll be discussing. We will be discussing about the important activities that you have in your textbook and different chapters of biology. Along with that, as ma'am just mentioned, we will be discussing about the important diagrams also. So please make sure you stay with us. It's a very short session. The session will not be more than 45 minutes or half an hour so please make sure you stay with us. We will not be taking any extra time. We'll be jumping right on the topic and I'm leaving. I'll join later and over to you everyone. And ma'am.
Yes, so good evening everybody. Now a lot of you, yes, so today's class is going to be mainly like a few things that I would like to start out, right? So first and foremost, we are going to be looking at all the chapters that are there from biology, right? Now the Maha Marathon for biology is already done, so we will not go into detailed concepts because we have already taught that. The video is available on the channel. You can check it out in case if you are very new to class, right? Now secondly, the thing is, I want to make sure that all of you have gone through the questions because Ankita ma'am and I have solved the competency based questions as well. Now the other thing that is there is that in today's class, what are we going to do? So what are we doing today, right? So what are we going to do today is what I'm going to quickly write. So first and foremost, everybody go and get your NCERT textbooks with you, right? Go and take your NCERT textbooks and make sure you have a pencil with you so that you can make a note of all the important pointers, right? So go quickly catch hold of your NCERT textbook and pencil so that we can get started, right? Now the second thing that we're going to do, yes? So the second thing that we're going to do is we will look at all the important activities, okay? Mind you, I am saying the important ones. There are some that are not going to be important on an examination front, right? So please make sure that we do the important activity ones, okay? We are only doing those. And third and foremost, along with this, what we will also do is we will mark the important diagrams that are needed on an examination front, right? Now on an examination front, there are chances that some may come for drawing, right? While some will only come for labeling or you will get it for observation based, right? So here we will go through all the diagrams from NCERT and we are going to get started. Now I know somebody is telling me, ma'am, आपने question number 5 नहीं किया control coordination से, मैं जरूर करूँगी और Telegram पे मैं डालूंगी, right? Right now is not the time for me to do it. So everybody, let's get started. We'll not waste too much time, but I hope in the meanwhile, all of you hit the like button on this video, right? Yes. And everyone, whoever is going to draw diagrams, no drawing with pen Akansh, only with pencil. Yes? So shall we get started? Can I have a quick Josh level check on the chat? And let's get started, okay? Give me a quick thumbs up. Ma'am, can we color diagrams? Don't color diagrams and all. No time. You will not have time to do all that. So, might as well not, right? And whoever's having random conversations, please make sure that you all focus, right? Stay focused in class. Do not have random conversations. So, let's get started, everybody, with life processes, right? So, life process may everybody go to your page number. I'm assuming it's 101, I guess, right? So life process is there in page number, oh, I'm not able to find the page number for this. Yeah, 93, go to page number 93. This is not a mentee, right? Everybody go to page number 93. Are we all there in page number 93? Okay. Ashok, if you're, Yuvan, if you're starting biology, you can watch the Maha Marathon, start with life processes, right? Okay. Now as we go down, we're going to scroll through pages, right? So as you all know, here when we come here, first diagram that we're going to encounter is in page number 96, right? Now page number 96 pe we have an activity and we have a diagram also, okay? Now see, in your sample paper, cross section of leaf has come, right? We have seen cross section of leaf has come for this. So learn this, not to draw, only for observing and labeling, right? So make sure that you learn these two diagrams. Mark this. Yes, make sure you make a tick mark saying labeling can come or observation can come. Sample paper pe this has come, right? So keeping this in mind, we are going to go and have a look at the first activity which is there on your screens. It's right there on the same page, so let's solve it together, right? So now here the question is very simple. There we have taken variegated leaves, right? So we have taken a potted plant with variegated leaves. Yes. So what are variegated leaves? Can you all tell me? What are variegated leaves in the meanwhile? Yes, I need the answers. What do we mean by variegated leaves? So I will go ahead with the question, but I hope you all know, right? Now when you talk about we've taken some variegated leaves, yes, they are colorful leaves which are there, right? So now those of you who don't know, they have colored parts. So there would be some green parts, then they may have red, or they may have some, they will have some maybe, yellow color part, right? Patchy leaves. Yes, simple way of doing this, right? So now we know that what we have done is we have taken a variegated leaf or a plant which has variegated leaf, and we have kept the plant in a dark room for 3 days, okay? So that all the starch gets used up. Now this process is what we call as destarching. What do we mean by destarching? Destarching is a process wherein we see that all the starch gets used up. And why do we want all the starch to get used up? So that at the end of the day, we know that it will not interfere with our process, right? So everybody, those of you who have doubts, right? Let me tell you, I will take it, but first listen to the explanation. Then I am going to take doubts. Then only this is going to be a fruitful session. So listen to me. Now what have we done? We have taken a variegated leaf, put it in dark room for destarching, right? Now we have kept this plant in sunlight for about, after destarching, why should we destarch? So that all the starch gets used up, right? We don't want the starch to interfere with the result, right? Existing starch that is there. We want to start it from scratch, right? We need to have a proper experiment. So in this case, what we have done, we have kept it for destarching, and then after that we have gone back and kept it in sunlight for 6 hours. Blessing, Priya will be your friend as long as you pay attention in class. So I need everybody to pay attention, okay? Now after keeping it in sunlight for 6 hours, what I have done, I have taken one leaf and I marked all my green parts. Because remember, we know that variegated means there are colors, right? So we know that there are different colors which are there. So I have marked all my green parts, okay? And then I kept it in hot boiling water for few minutes, yes? And here I kept it in boiling water and then I kept it in alcohol, okay? And then after that I heated it till, like in a double boiler, till I boiled it for some time. Now our first question is question number 1. What happens to the color of the leaf? And what is the color of the solution? So can you all tell me what is going to happen to the color of the leaf? Yes? Everyone, I have destarched it, plucked one leaf, put it in boiling water, then I am going to put it in alcohol. Everybody is telling me, ma'am, it will become blue-black. But is it going to become blue-black? Color changes, okay, blue-black, everybody is telling me blue-black. See, blue-black will happen when iodine comes into the picture. Yes, Apurva, it will get decolorized, it's going to lose its color. So in this case, what is the answer? Leaf will become decolorized, right? Decolorized. It will lose its color. It will lose its basic color that is there. So whatever chlorophyll was there, all of that is gone now. And where does this color go to? It goes to the solution. So what is the color of the solution? It will be, greenish in color, right? So we see that in this case, the solution is going to be greenish in color. So now I have a decolorized solution that is there, right? Now, decolorized leaf. Now I'm going to take this decolorized leaf which has lost all of its chlorophyll, and then I'm going to dip it in iodine solution for a few minutes. Okay, I'm going to dip it in iodine solution, and then I'm going to take it off and I'm going to observe, right? So now what will I observe after I dip it in iodine solution? Crotons is an example, that's all, right? It's an example of a leaf. What am I going to observe? Somebody is saying, ma'am, no color change, blue, black. Now see, your answers are correct, but here and there are some mistakes, okay? Blue, exactly, Lakshita. Starch present areas, yes. So starch present areas will show blue, black, right? So basically, wherever starch was there or wherever green parts, you remember we labeled all the green parts, that because in a variegated leaf you will have green and colored parts, right? So हमने green parts label कर दिया और colored parts भी label कर दिया. So whichever were originally green, very good Suresh, exactly. So all the parts, right, which were green in color, right? So I'm going to write this down. Parts which were originally green in color, we see that they will turn blue black, right? Because whichever was blue, was initially green in color, they could photosynthesize. That means starch formation is there in those areas, and when you do this, when you add iodine solution to it, it is going to turn blue black, right? So that is why in this case we see that those parts are going to turn blue black as per figure 6.2. Yes? And what can you conclude about the presence of starch in various areas of the leaf? What can you conclude? Can you all tell me in the chat very quickly? What can you conclude? Exactly, Priyanka. The part which was not green will not change color. Okay? Yes, I will repeat this once again. Okay, ma'am, is it important to write option in answer sheet? Of course. See, like I said, no. Mainly, you need to make sure when you're writing the answer for MCQ, write option A, B, C. You will have to write that part. Exactly. Very good. This detects the presence of starch, right? So basically in all these parts, we observe that there is presence of starch, okay? So now quickly, what did I do? Once again, I took a plant, I de-starched it, kept it in dark room, okay? And this plant had variegated leaf, right? Now from here, of course, what will we do? We took the variegated leaf, we put it in boiling water, got rid of the chlorophyll, we put it in alcohol, decolorized it, got rid of the chlorophyll, and then I did my iodine test. Why should I do iodine test, or why am I adding iodine? Because iodine reacts with starch to give me a complex called the iodine starch complex that is blue black in color, right? That's why I am adding iodine solution. It acts as a stain, right? So which is why in this case this activity is important. Now for those of you who are asking me, ma'am, will activities come? They can come for MCQ. If you have seen your sample paper, practice paper, you will get this, right? So it's important that you go about it, okay? And you understand this. Now next up for diagram, going on to next page, right? If we move on to the next page, we are in page number 96, right? I hope all of you are writing the answers along with me in your textbook. That's why I asked you to get your textbooks and come. Why alcohol? Because alcohol will get rid of the chlorophyll that is there, right? That's why we are doing it in alcohol. Now moving on. Make sure you learn the diagram for open and closed stomatal pore, right? Very easy diagram. They may not ask you to draw per se, but it's good to draw and keep, right? So keep a note of this particular diagram as well. Now we will move on to activity 6.2, which we will discuss. Now 6.2 is very important, okay? Please make sure you make a star mark here. 6.2 is very, very important, right? Now in this case, right, we know that in 6.2, we have taken two plants, okay, two very healthy plants which are of the same size, right? We have taken two plants which are of the same size, and then we kept it in a dark room for 3 days. Now what did I do? I kept each plant, so let's call this as plant 1, right? And let's call this as plant 2, right? I'll take your doubts, bacha, but I need you all to pay attention to this, okay? Now I've kept plant 1 and plant 2 in a dark room, right? Now what did I do? Now I placed a separate glass plate on each of it, right? I kept one glass plate. Okay, now what did I do? In plant 1, I put a glass plate that has potassium hydroxide. Now remember this. Potassium hydroxide is used to absorb carbon dioxide. This is very, very important because we had a question in the practice paper also, right? So this is also very important. Now moving on, what did we do? We covered both plant 1 and plant 2 with bell jars, and then we sealed the bottom with Vaseline so that no air will enter, right? Now air cannot enter. Now what has happened? I kept my plants in sunlight for 2 hours, right? And after keeping it for 2 hours, I took leaf from here. So I took leaf 1 from plant 1 and I took leaf 2 from plant 2, right? And I did a presence for starch. Which leaf is going to turn blue-black in color? Will it be leaf 1 or leaf 2? In the chat, which one will show or turn blue-black in color? Leaf 1 or leaf 2? Yes, or you can say plant 1 or 2, anything is fine, right? Okay. Yes, very good. Leaf 2 is going to turn, but you also need to give me a reason, right? Why is leaf 2 turning, right? That is what they are asking. Ma'am, don't know. It's okay. Right? Yes. Okay, somebody's asking me, ma'am, what is the purpose of this experiment? I will answer that as well, right? Exactly. See, now when I kept it in the dark room, we have de-starched it here, right? We have de-starched the plant. Yes, that means no more starch is left. Now when I wanted to carry out my experiment, I kept potassium hydroxide in leaf 1, and this is said to absorb carbon dioxide, and we know that carbon dioxide is a raw material. No, carbon dioxide is a raw material for photosynthesis. So if all the carbon dioxide is absorbed, then how will this plant do photosynthesis? It will not be able to do photosynthesis, right? But in this case, carbon dioxide is still available, which means photosynthesis will happen, starch formation will take place, and leaf 2 here is going to turn blue-black in color, while leaf 1 will not turn blue-black in color, right? Now in this case, what are they asking? What can you conclude from this, right? So they're asking us, what can we conclude from this experiment? So can you tell me what we can conclude? Ma'am, will this activity come? They will ask you a modified version of this activity. If you remember, they gave you one with plant, living plant, dead plant. I think in your practice paper it was there, right? So in this case, this is very, very important. This activity is important. Please star mark this. Exactly. The inference is that carbon dioxide is necessary for photosynthesis. So now one thing, let me tell you with activity based questions, okay, and I want all of you to hear me out. Now purpose of 6.1 is to tell us that chlorophyll or regions of the leaf which have chlorophyll is essential, or chlorophyll is essential for photosynthesis. And what is the purpose of 6.2? A purpose of activity 6.2 is to tell us that carbon dioxide is necessary for photosynthesis, right? So this right here is very, very important. Try to understand the inference, and this is very easy, right? This is going to be very, very easy, right? If you get activity based questions, MCQ pe to zarur it will come, so be very, very careful. So far so good. Give me thumbs up in the chat. Jaldi, 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 right? Okay. Harsh, what is it? What is it that you want? Unnecessarily do not, right? Please do not come with random comments in the chat, right? So again, like I said, if you don't want to be here, you are always welcome to leave. A lot of my students really want to learn from me, so let them learn. Don't unnecessarily talk about it. Okay, so now everybody quickly telling you, right? 6.1 mein we did with variegated leaf. 6.2 mein we have taken potassium hydroxide, right? So in this particular one, we know that in potassium hydroxide, basically we have kept it inside a bell jar to absorb all the carbon dioxide, and we know that photosynthesis will not happen if carbon dioxide is not there. But on the other hand, it's a control here, which is why carbon dioxide will be there. Yes, 6.1 diagram. Oh God, which one was that? 6.1 diagram. I have already marked 6.1. Yes, you need to learn this for MCQ because in your sample paper it came. So they'll ask you, right, in which region will photosynthesis takes place, region 1, region 2. So they may ask you all of this, right? Ma'am, where are epidermal cells present? See, these are your epidermal cells, right? These regions are your epidermal cells, okay? These two regions, I hope you can make a note of it, right? Now let's quickly move on, right? Ankita ma'am also has to come, do a lot of it. Let's move on. Now let's go on to figure number 6.5, nutrition in amoeba. Nutrition and amoeba, chances of it coming to draw are very slim, right? But even if they ask you to draw, it's going to be very easy. So just make a note of this, right? It may come for labeling, right? Or it may come to draw, but this is like a 40 to 20% chance that it will come, okay? So make a note of this as well. Now let's move on further down to activity 6.3, which is a very interesting activity, right? Ma'am, how many times should we learn to draw? Once. Amoeba ka diagram is very easy, right? Ma'am, it will come for theory question. Maybe it can come, right? So we're going to do all activities from NCERT, Anshu. So let's get paced up. Let's pace it up quickly. This is a very interesting one, okay? Now, if you look at this particular one, we have two test tubes which are given to us, okay? You have test tube A and you have test tube B. Now in both test tubes, what we have done is we have added 1% of starch, right? We have added 1% starch in both of test tubes. We have put two solutions, okay? Then what did we do? To test tube A, I also added 1 ml of saliva, okay? I added 1 ml of saliva, and in this case I left it like this for 20 minutes, right? I kept it for 20 minutes and I left my test tubes as it is. Now after this, what did I do? I added iodine solutions to both of this, right? So I added iodine solution to both of my test tubes, right? Now after adding iodine solution to both, yes, which test tube will we observe color change? Test tube A or test tube B? Can you tell me? Ma'am, can we draw diagrams in questions where they have not asked? Only if it is supporting the answer, you draw it. Only if you are confident about it. Don't unnecessarily draw it, right? Yes. Everybody's telling me, ma'am, test tube B, we will observe color change, right? Now, what does this indicate about presence or absence in starch in both the test tubes? So, clearly, if color change is there in test tube B, there is presence of starch, right? So, there is presence of starch. But what about test tube A? What happened in test tube A? Can you tell me? Yes? What has happened in test tube A? Jaldi, jaldi. Let's pace it up. Yes, important diagrams I am explaining, Leah. Just pay attention. Very good. Starch is digested or salivary amylase acts on it, right? So here we know that there has been digestion of starch, right? Now what does this also mean? What does this tell about the action of saliva? We know that action of saliva here is saliva has salivary amylase, right? So we see that there is salivary amylase which acts upon the starch that breaks it down into simpler forms, right? So because of this, we see that there is only color change in B. So quickly, we have taken same amount of starch in both and we have added only 1 ml of saliva in A. And we know saliva has salivary amylase that will act on the starch, right? So when it has acted on the starch, there is no longer starch present in this. Something else, it has become into maltose, right? So as you know, it has become into maltose. That means when I add iodine solution, color change will happen only if starch is present, right? Which is why in this case, we see that color change is observed in B. What does this tell us? Saliva that is there, it acts on starch or facilitates the digestion of starch, okay? So I hope all of you have made a note of this. Yes, all of you have made a note. Important diagram मैं बता रही हूँ, आप क्लास में अटेम्प्ट फोकस कीजिए रिया, आप सबको मिल जाएगा, ठीक है? Give me a thumbs up and we're moving on. All right, okay. Now let's move on. See, human digestive system is not going to come for drawing. Labeling is important, right? It can come for diagram based question. Please make a note, right? It is only going to come for labeling. So now let's quickly move on to the next one. Okay, let's go down all the way till activity 6.4. Yes? Activity 6.4. Very easy one. Okay, super duper easy. We can finish it like this. I have taken two test tubes A and B. Okay, I have taken two test tubes A and B, and I put lime water in both A and B, right? I put lime water in both A and B. Now what did I do? I had somebody blow into the test tube in one, and I took a pitchkari and then I was pressing it like this in the second one, right? Now we saw that in B there was color change, right? It turned lime water, or the lime water here became milky. But in this case, we saw that it remained as it is, or we did not see a big change, right? So we see that the change was relatively little, right? So we see that it was relatively little. So what does this tell about the carbon dioxide in the air that we breathe out? What does this say? Yes, carbon dioxide is there. Carbon dioxide is turning it milky. But it is based on how much of carbon dioxide? Exactly, Divya. Very good, right? So now we know that mostly, right? In most cases, more amount. As a matter of fact, if 0.04% of carbon dioxide is present in the atmosphere, we see that we breathe out or we exhale out actually more than 100%. That means almost 4% carbon dioxide, 100 times more, right? That means that we breathe out 4% of carbon dioxide, which is why in this case we see that when we actually blow into it, we see that it turns milky. Ma'am, what if we kept carbon dioxide in pitchkari? Yeah, obviously, if you put only carbon dioxide, it will turn milky, right? Now, for those of you who are asking, ma'am, is our lecture enough for boards? Pooja, see, you'll have to read your session, right? But I hope that all of you that are there, I mean, you'll find our sessions helpful, but go through your NCERT. Ma'am, respiration means carbon dioxide. Somebody's asking me something. Respiration means carbohydrates are broken down to form glucose. No, that is digestion. Carbohydrates getting broken down into simplest form is digestion. Okay, ATP part Garvit Raj, I'll explain. Liver helps in production of albumin protein. No, Nabija, that's not part of your exam. You don't need to worry. Okay. Ma'am, I have doubt in 6.3. You can ask me, but I explained 6.3 in detail. So whatever specific doubt you have, you can ask me. Yes, I will mainly teach in English only, so that I'll already give you a heads up now. Now moving on, 6.5 is very simple, right? Very, very easy part. Now listen, students. I am explaining so slowly to a point that if you listen to me, you will know the answer. Okay, but if you keep typing and texting in between, you might lose out on what I'm trying to explain, right? So everybody, next one is very simple. You have a box, you have put sugar solution in it. Then you added yeast also, right? You made a hole and then you put a bent U-tube and then I took this and then I put it in lime water, right? I put it in lime water. Now what are they saying? They're asking us what will happen to this lime water. Can you tell me? So I took a sugar solution, added some yeast, mixed it nicely and then I connected one tube. I closed everything and then I connected it to lime water, right? What are we going to get? Quickly, lime water is going to turn milky. Exactly. So what will happen here? Lime water is going to turn milky. So what does this tell about the end products of fermentation? Or another way is end products of anaerobic respiration. Yes, end products of anaerobic respiration in yeast. What are the end products? Jaldi, jaldi. Very good. I can see the answers. It is going to be carbon dioxide. Right, so we have ethanol and we have carbon dioxide, right? So we see that these are the two end products. Very good, very good. Easy peasy experiment, no, super easy. All you need to do is to break down the information that is given to you, right? Ma'am, I am not able to understand this activity. Yuvan, we took one glass, we put some sugar in it, sugar solution or fruit juice. I put yeast in it and we know that yeast will ferment. It will do anaerobic respiration and it will break down the sugar, giving you carbon dioxide and ethanol. So if all of you have not studied life processes so far, first I'll say you go study life process, study the chapters and then do the activity. Because if you have not studied earlier and you're coming to for the activity session, you will not understand a lot of things, right? So that is why I am stressing on the fact that you should first study your concepts. We could have easily done this yesterday when we did the marathon, but we didn't do it because you should first understand concept. Only then you will be able to answer these questions, right? Okay, Soumya, I'm happy that it's going to help you, right? How long does it take? It takes about 15-20 minutes, right? Well, I mean, or half an hour. Depends from the concentration of sugar on multiple factors. Why sugar? Because sugar. Why sugar? Because at the end of the day, to get energy, sugar is needed, right? Can we expect this for subjective questions? You can. 2 marks for sure. Competency, these are also type of competency based questions which are there. Now I'm sure diagram wise Ankita ma'am would have told you that this diagram is not necessary for drawing or anything, but I'm sure yesterday Ankita ma'am told you that to study respiration this is very important, right? Ankita ma'am has explained this in great detail. Please make sure that you are going through when you are revising respiration. Now quickly going on. See, ATP ka, this will not come, right ma'am? This will most likely not come in detail, so you do not have to worry about it. Yes, you can just read it for understanding. Now see, activity 6.6, no need. It's not, you have to sit and count in aquarium and all, you don't need to know about this, right? No need, activity 6.6. Now moving on, let's go down here to this particular diagram, which is human respiratory system. Respiratory system again only will mainly come for labeling, right? Mainly it will come for labeling, right? But if you want to learn to draw also, because maybe way back in 2012, 2018, once or twice over a period of many years, here and there they may ask you to draw, but chances are very less, right? Okay, why are there saponification? You can ask Kriti ma'am, right? Soap formation and everything, you can talk to Kriti ma'am. Yahan pe activity 6.7 also nothing to worry about, right? Because here it's all observation based. So this right here is not going to be very important. Orange box in some places are for skimming, some are important. So read it. Everybody's most famous question, ma'am, heart ka diagram aayega ya only labeling? What is the scene with heart ka diagram? So let's understand this, that heart ka diagram will again come for labeling, right? They can ask you a diagram based question. Please mark this in your book, right? It will only come for diagram based question, right? Ma'am, pakka ho gaya heart? Amazing. Most often than not, they will not ask you, right? See, in the sense when I say diagram based question, they'll say A, B, C, right? And then they'll ask you identify which part is this, which part is that. They'll ask you all of those things. Everybody asking me content related doubts, please go back to our Maha Marathon and check it out. We have answered all the questions which are coming right now. I am not taking it because if I start teaching, the session is going to go longer than expected. See, this is double circulation ka diagram. Now we told you that when questions come on double circulation, you don't need to really draw this, but you can draw a flow chart, right? A flow chart diagram is enough, but to draw a flow chart, you should know the labelings from this particular thing, right? So everybody, please make sure that you make a note of it and draw the flow chart for this. Are you all with me? Are you all with me? Thumbs up in the chat, everybody. Are you all marking it? Bachas, double circulation question will come. Chances of it coming are high, right? But schematic diagram is enough. Okay, they'll not ask you to waste your time drawing all of this, right? Blood pressure is important. Okay, somebody asking me, ma'am, I'll not read orange box. Blood pressure is important. Please go through this. Okay. Now going niche, we have one activity here which is a very simple one, right? Activity 6.8 is important, which is based on transpiration. Gia, I have drawn all this. Please go to the Maha Marathon session and check it out. I am not going to do anything that I've already done earlier. Lymph I have explained detail in Maha Marathon. All of you go watch that session. It will definitely help you out. You won Maha Marathon session. Go check all this out, right? You will definitely find whatever you want. Yes, all of that we have explained in great detail. Aram se we have explained. You will find the answers there, right? Is lymphatic system important? It is important. Okay? Now here you have one experiment where you have a plant. Okay? You have one plant here and you have another plant here. Right? So in one I have taken a plant with leaves and in another one I took a stick of the same height. Now I covered both the soils with plastic sheet so that evaporation does not happen. Then what did I do? I covered the whole thing. Okay, I covered the whole plant with plastic, right? So let me erase this. I covered both of this with plastic wrapper. Now, and I've kept it outside in the sun for some time. What will I observe? I have A and I have B. What am I going to observe in A? Can you tell me? Yes? Can you tell me what will I observe in A? Yes? Exactly, Sayed. In practice paper, it was a mix of 6.8 and 6.2. Exactly. So that's why I said 6.8 is very, very important, right? Super duper important activity. Pay attention. Very good. So on the surface of A, I will observe water droplets. Can you tell me why we will observe water droplets? Yes? Ma'am, do we draw? I'm drawing this for your understanding and for you all to understand faster, right? So that is why I'm doing it. Okay. This can come in MCQs. Mostly MCQs that are there will come in this format only, right? Very good. It is because of transpiration, right? Transpiration is the reason. But in B, no plants are there and no evaporation will take place. So here we see that there will be no droplets. Yes? Very good. Very good. So now most of you have got activity 6.8 also. So well done all of you. Most of it of life process is done. I think in another 5 minutes we are going to wind up life process, right? I mean control coordination as well. We will wind it up very soon. Now going down, learn this diagram. It's not like it will come, but sometimes it may come to draw, right? Chances are that it may come to draw. So learn the labeling for sure, right? Very, very important. Learn it for diagram as well. They may ask you. Yes, they may ask you on this. Now going down, right? If you go, nephron diagram is there. Learn the nephron diagram. Labeling is also very, very important. Yesterday when Ankita ma'am was taking competency based questions, we had a nephron based diagram, right? And then along with that, if you see earlier on, also we see that nephron based diagrams can come. So this is very, very important. Diagram may come. Right, like I said, chances of drawing may not be that much, but nonetheless it's important to learn, right? Nephron diagram is important, so please put a star mark. Okay, star mark. Artificial kidney, not very important, but read through it. They can ask you for MCQs, so read this. Okay, super important. Urine formation, very important hai for those of you asking me. So with that everyone, we are done with life process. Are we ready? We are done with life process. Are you all feeling confident? Are you all feeling super confident? See 6.8 pe bacha, what did I do? I took one plant, I had plant in it. I took another one with only stick, right? I covered the soil so that evaporation doesn't take place, and then I covered my leaves with plastic. Yes, I covered it with plastic. Then because transpiration takes place, we observe droplets, right? So that is the intent of activity 6.8. Okay. Must you know boxy? Go through all the questions, but don't take too much tension, right? Don't take too much tension. How do I get out of full screen? Oh yeah. All right, so quickly we are going to move into control and coordination. Control coordination, very, very easy, and then I'm going to hand it over to Ankita ma'am, right? Jaldi. Everybody hit the like button also. That's important, right? So now let's move on. Okay, but before I go ahead with that. All right, I hope all of you are able to see my screen right now. Yes, I hope my screen is visible. Now here in control coordination, activities are not much, okay? They are very easy only, right? Very simple it is. Yes, for those of you who are still a little confused with the activities, you can rewind back and check. I've gone a little bit in detail only. You can check it out. If not, don't worry, I'll discuss it later, right? So now here you have neuron diagram. Neuron diagram can come for drawing, right? And neuron diagram is very easy. You have nothing to worry about also. We have done neuron multiple times. So make a note of it. This can come for drawing. Yes, to draw. And it can come, you know, maybe diagram based. So labeling is also very, very important. Put a star mark here. Yes, everybody put a star mark. Now moving on, niche, see this will not come. Neuromuscular junction will not come for drawing, so don't take tension, right? 7.1 is just for you to have an understanding of what are receptors, but you don't need to look at 7.1, right? They're asking you to close your nose, open your nose. They'll not give you questions like this. Then when you go niche, reflex arc, diagram is important. Okay, labeling is important. All of you learned this, okay? Very, very important. I'm sure when Ankita ma'am took class yesterday also, she would have stressed on how important reflex arc is, right? So everybody put a star mark and make sure that reflex arc diagram is marked. Yes? Everybody quickly in the chat, you have to keep telling me thumbs up as we switch to the next one, okay? As and when we go, you are giving me thumbs ups in the chat. They may ask you to draw. If it comes for 4 marks or 3 marks, diagram supporting your answer will be good. Now brain ka diagram, everybody's most biggest stress of life, right? Oh ma'am, brain ka diagram aayega. No, Okay, learn labeling. Yes, learn the labelings. Most often than not, they will not ask you to draw the brain diagram, right? But even if they ask you, it is very simple only, right? I know it looks very complicated, but most often than not, they may not ask you to draw brain diagram, but they will ask you diagram based questions, right? So diagram based questions they will ask you. So everybody, please make sure all the labelings you learn properly. Proper you will have to come, okay? Properly learn this. Now going niche very quickly. Here in this case, two things, right? Geotropism, plant showing geotropism, you need to learn this particular one, right? So here you will have to learn this particular diagram on plant showing geotropism. You will get diagram based question. Yes, you will have a diagram based question on this, right? All right. Ma'am, I'm not understanding what you are trying to ask me. Oh, okay. If there's something that you are not able to understand, don't worry. I'll just ask me once again. Now, activity 7.2 can come in your exam, and we've already discussed this. I took one plant, I put it in conical flask, then I covered and I kept it near window, right? And then what did I do? I saw that the old parts of shoot and root have changed direction, right? So here if you see, I have kept there has been a change in shoot and root direction. So now you should tell me what and how are the difference, so basically you have to tell me why this is happening, right? Why is this happening? Can you tell me? Why is this happening? Ma'am, 7.6. It will not come for drawing. It will not come. Exactly. Yes. This is all about, I've simplified the activity question, right? So this is also, there are two answers. Phototropism and auxin, right? Because we know that auxin deposition happens in the shady region, right? And of course, as a result, it causes bending towards stimuli because auxin is sensitive, right? It is sensitive to light. So auxin accumulation happens in the opposite direction. Yes? So this is the main thing what you can conclude from this particular activity. Very easy. Super duper easy. Now with this, of course, everybody, we are done, right? Now here 7.3 is identify endocrine glands mentioned in the figure, and very easy. So here also labeling is important, right? They can ask you this. Labeling, learn the labeling and you can get diagram based questions. Yes, but chances of this coming in are very less, right? So this is very, very less that they will ask you this, but don't take tension about it. Learn the important labelings that are there, right? Haan, pre-board mein aaya tha. Ma'am, can we do flowchart for reflex arc? You can, but if they ask you to draw the reflex arc, then you may have to learn it, right? So it's very simple. Draw one finger, draw just the pathway like this, and draw one, like a X-like thing which shows the spinal cord. That's all. And I'll tag some of the videos in the comments of this session where I have told you and given you tips and tricks on how to draw reflex arc and everything. So I will pin those videos, right? I will pin those videos in the comments of this thing so that you will be able to understand. Yes? All right. So with this, of course, everybody, we are done with control and coordination. We have had a look at all the important questions, and we have had a look at all the activities as well. So I hope now all of you are feeling confident about this. I hope you are all more confident. Yes? Give me a quick thumbs up, then we will call in Ankita ma'am to take us through last two chapters. Okay? Very simple and easy. Nephron diagram, learn for drawing and labeling. Both. Okay, yes. Those of you who have issues with my volume, you can reduce the volume in your phones, right? Yes, all right, everyone. Ma'am, what's happening in the roots? Geotropism is happening in the roots, right? Very good. So now let's all call Ankita ma'am, and before that, I hope all of you have hit the like button on this video. I need to have at least 900 likes on this video, right? So everyone, after this, please go ahead and watch the Maha Marathon where we have taught you in detail, right? And then go back and try to solve it. And don't get worried. Exam is going to be easy, bacha. Very, very easy it is going to be. So don't worry, okay? So let's call Ankita ma'am now.
Yes.
Yes, ma'am.
And they can hit the like button.
Over to you.
Thank you, ma'am. Hello, everyone. Good evening, everyone. Good evening. Careful. Awesome, everyone. So let's get started, right? I'll just increase the height of the table and then we'll move ahead. So I hope that all of you have the clarity, right? That all the important diagrams, right? I hope that you're keeping a track of all the important things, right? See, again, as Ashwara ma'am was telling you, please make sure if you have any doubt, go and watch the marathon sessions. Those sessions will be helping you throughout, okay? Especially for biology, because we have gone into the detail. Now we are starting with the how do organism reproduce, a very, very easy question. The diagrams that we have. So here we have, right, let me just, I think this is fine. Increase a little bit. So here we go, everyone. Yes, hello everyone. So I want all of your attention now. I want all of you to pay attention so that we can quickly finish this off and then you can go ahead and take a break, because after this class we will be having one more class of physics. So please make sure everyone you take a good break after this class and come back again. So let's get started. So this is a very interesting and super easy chapter, which is how do organisms reproduce, right? Now, in this particular chapter, what are important diagrams? We'll be looking at that. Now, in this particular chapter, if you talk about the activities, so the activities are not that directed, right? They're more of theory based. So let's take a look over here, the activity that we have, which is activity 8.1. And in this particular activity, what are we discussing? We are discussing about the different modes of reproduction that we have in single cell organism or unicellular organism. So here we have that we will be taking, a small amount of sugar and we'll be adding into the 100 milliliter of water. And you know what? They are adding the yeast. So they are adding the yeast, right? And after some time, they will be observing the yeast, right? And they will be observing it under the microscopes. So of course, we know that sugar will be helping the yeast to grow, and we will be seeing which process. Very good, Padma. Riya, yes, if you pay attention, right, and if you watch the videos that we are doing recently where we have discussed about the marathon session, if you have the clarity, if you have read NCERT, you will definitely score more than 95. Very good, everyone. So what process is happening over here? Budding. So chances of these activity coming from how do organism reproduce are very less. Okay, so don't think about much. You should know the different types of asexual reproduction modes that yesterday when Ashwara ma'am took the class, ma'am have gone into the details and have discussed about these. So please make sure you watch that video, right? Talking in terms of the next activity that we have over here, let's take a look. So here we have the next activity which talks about the spore formation, right? About the spore formation. Yes, this chapter is very important from the exam point of view. Yes, here we have, see, what are they doing? Basically, they're asking us to take a slice of a bread, right? Then they're asking us to keep it in a moist condition for some time, and then we will be able to see the growth of fungus, right? And which fungus? Of course, we have mold, right? Or in the scientific name, we call it as rhizopus. Very good, right? So over here, we will be able to see it. Okay, this is what the activity is all about. Yes, we'll see the spore formation. So then, of course, you can mention about the activities. Okay, you can mention about the rhizopus over here, right? You have the bread wala. Yeah, this activity is a little bit long over here. Yes. Okay, this one. Clear, everyone? Chaliye. Now let's go back over here and let's discuss about the important diagrams. So luckily in how do organisms reproduce, right? We have very easy diagram. So these diagrams can come for you to identify, not for you to draw in the examination, but to identify. By looking at these, and then they can ask you to identify the type of asexual reproduction that we can see in the organism. So please make sure, everyone, that you go through the diagrams that you have in your NCERT textbook. So over here we have the binary fission in amoeba. Here we have the same in the leishmania, so please make sure you take a note of this. Then if we talk about the other diagrams, like we have the multiple fission, remember the name, plasmodium, right? Then of course we have the regeneration in planaria, we have budding in hydra, and we have the leaf, right? Bryophyllum. So all of these diagrams, right? Yes, all of these diagrams are very important. Nobody will be asking you to draw these diagrams in the examination, but you can have the image based question on this. So please make sure you go through these. Okay. Now let's talk about the activity number 8.3, which is of course again they're asking us to observe. Yes. See, as of now, if you look at the trend, the CBSE is not asking you to draw the diagrams in the examination. Okay? You can get full marks in science if you pay attention, and if you have studied everything, nothing is stopping you. We will discuss about the variation after some time. Wait for some time, bache. Good. See, what this activity is about, basically we have to observe a permanent slide of amoeba, and then we have to observe one more slide where we can see the binary fission. So we can definitely write that the daughter cells, right, will be very similar to the mother, right, or the parent cell. Very easy, nothing much over here for all of you to worry about. I'm sure by now you all are the master of this particular chapter. Okay. Here everyone, we have 8.4 activity, again a very simple activity. What the activity is asking us to do is go to the pond or a lake. We have to get the dark green color ka that layer that we have on the water, right? They're asking us to collect that. Basically it will have some filamentous structure. Yes, and they have to identify. We know that, right? We will have the Spirogyra, that is an algae, and it reproduces by using which method? Can you quickly tell me, everyone? I will be checking your knowledge. Right? Spirogyra reproduce by using which method? Very good. Yeast culture. The growth of the yeast. Very good, everyone. It is called as the fragmentation. MCQs for the marathon, we already did the questions, right? The important NCERT questions of biology. You can please go and check it on the channel. Ma'am, what if the only exercise question? Yeah, you can do that also. But if you want to do that, please make sure you're reading the NCERT. Good, everyone. The process is the fragmentation. Okay, let's take a look at the next activity. Over here, we have the plain India diagram. I already said it's important. It can come for the identification. Now, this is everyone important, which is just talking about the vegetative propagation. Everyone, can you quickly tell me what is vegetative propagation? Microsoft Tech Expert, we have already discussed the NCERT questions for biology and for other subjects also. So if you go on the channel, very recently we did that. Yes, you will have everything. Yes, thank you Yuvan for spreading the positivity amongst your friends. Everyone, what is vegetative propagation? Very good. So of course, when the vegetative parts are used to reproduce, right? Of course, these parts will be helping us to have the new plants, right? They actually help in the production of the new plants. We call it as a vegetative propagation. So out of that, of course, we have root, stem and leaves. In this particular activity, they're asking you to cut the potatoes, right, into small pieces, keep it on a wet cotton, right? And they're asking you to have potatoes which have the notch or the buds. I'm sure you would have seen them at your home. So basically you have to cut them, spread them on the cotton sheet or a cotton, right, and have some water. Basically that water will be helping to provide the moisture that will be eventually helping the potato slices to grow further, right? So you will see after some time you will be able to see some growth. You can try this activity, but after your examination, this activity is not that important, so please do it during your summer vacations. But you should know what is happening over here. Cool. Let's take a look at the next activity, everyone, which is activity number 8.6. Very easy. How many of you have the, I'm sure it's a very common plant around our surrounding, money plant? How many of you have money plant at your home? Very simple activity. Yeah, right. If you cut a small part of the money plant, if you add into water or into soil, eventually we'll see that it's growing, right? So this activity is about that only. So don't worry, you don't have to worry about this. Let it be. Let it go. Okay? Good. Now this tissue culture is important. Yesterday when ma'am had a class with you, right, Ashwara ma'am have taught you this in detail. So please make sure you go and watch it. Okay? This part is only important, right? It actually helps us to understand that how the tissue culture is happening. So please make sure you go and watch that part also. Good. right? Then of course we have the spore formation. We know that spore formation is very important. Of course, this is the spore formation occurs in the rhizopus. We just had the discussion, right? Good, very good. So Ria, there's no as such weightage, sorry Kunal, there's no as such weightage for the activities, right? But they will be a part of your question paper itself. Okay, everyone. So with this, we're done with the asexual reproduction, and as you all know, in a sexual reproduction, three most important diagrams. First, of course, the cross-sectional of the flower. You should know the parts. You should know where the anther is, where the filament is, where the stigma, style and ovary is. Apart from this, right, what we have is very, very important. Now, I want all of you to pay attention to this diagram. Please make sure you are aware about this diagram, right? This diagram can come in your examination to identify the part. It came in the last year paper. So, please make sure you have the understanding about this diagram, okay? This is very important. And then we have chotu sa cute little seed ka diagram, basically germination. So this is also important, but more than that, remember the names, right? Remember the what is plumule, what is radical, all of these things, because this can come in the identification part. Clear, everyone? Give me a quick thumbs up. Just need 5 more minutes of your time, and then we'll be done with this. Clear? Good. After this, what we have? Easy peasy, we have the reproductive system of both male and female. Both are very important. You should know all the labeling, so please make sure you practice the labelings, right? Practice the labeling. Nobody will be asking you to draw these diagram in the examination, but identifying the parts and writing their function can definitely come in your examination. So please make sure, right, that you practice the labeling for both male and female reproductive system. With that, I think there is nothing more here. Like there's no other diagram, there are no other activities, and we are done with this chapter. So everyone, I hope that you are clear with this particular chapter activities and the diagrams. No, it's not important, Syed. They can ask you, they can show you the diagram and they can show the leaves. You know, that's the vegetative propagation in the bryophyllum. Why not exercise question? We are not doing exercise question because we have already done that. Today's session is only focusing on the activities and the important diagrams. You have tissue culture. Syed, it's a very simple thing you're asking. We just discussed, right? It was there in our syllabus. It is there in our syllabus. We have the table. We have that cute little orange box. Everyone, clear? Give me a quick thumbs up. And please make sure you say quickly, everyone. Yes, I have just now reproduction. Very lucky actually that we just have heredity here and not the evolution. Okay. This diagram is not important, right? It just talks about the variation. But please make sure you read the paragraphs that you have over here, right? Please make sure you read them, especially initial paragraphs, okay? Then there's one activity which is really very chotu, cute little activity that definitely we cannot do now, is that you roam around in your class. Look how many of your classmates have the earlobes attached and free earlobes. See, yesterday we did a question, we know that attached earlobe. See over here, these are the attached earlobe, this is recessive. And over here, free earlobe are dominating. Remember this, the question can come based upon this. Okay? So, attached earlobes are recessive in nature, whereas the free earlobes are dominant. We have a glitch. Is it, Shiva? Yes. There's a lag. Is it a lag, everyone? We'll just wait for some time. You can refresh your page. It's fine only from our side. The question can come based upon. And the next, how many people are? It's fine from my side, everyone. Yeah. No, it's no lag. No lag. Yes. Now it's not no lag. It's fine. Yes. Good. Very good. Fine now, right? Okay. So we have this, everyone, and of course, yeah, I'll tell you the page number because you are asking for that. The page number is 143. Okay? So 143 is a page number. Please make sure you go and watch this. Chaliye. Next, everyone, very quickly, let's take a look at the next activity. Okay. Yes. Here we have the next activity. Now, okay, wait, I think it went up. I think the system, yeah, there's no lag, everyone. Maybe the screen, right, the system that I'm using is a little bit slow. I can see that now. So it could be of that reason. There was no lag. Okay. Here we have this activity, everyone. So it's a basically a very easy experiment. So we have this, that in the figure over here, we have the F1 generation, right, which was heterozygous. And in the F2 generation, what we saw, the result was that 1:2:1, right, or the 3:1. Okay. So the question is, what experiment would we do to confirm that the F2 generation did have the 1:2:3 ratio? So we will do this experiment. We'll do the monohybrid cross, and that's how we'll be able to know this. So there's nothing much for you to worry about this particular activity. Good, very good, everyone. See, again, in this particular chapter, there's no important diagram. Punnett squares are important, so please make sure you practice the Punnett square for the monohybrid cross and the dihybrid cross. Okay, everyone? I want all of you to give me a quick thumbs up. Now I'll take a very small, chotu sa pyara sa question, which was, what is variation? Variation is nothing but the difference, right, that we see from one generation to the another. That's how I'll put so that it's easy for you to understand. We have a lot of changes, right? We are very different from our parents because of the variation. Gitika, exercise questions are important. Please make sure you go through them. Yes, Sayyid. 9.2 activities is basically the whole chapter. Good, everyone. So with this, actually we are done, right? Please make sure, everyone, that you go through all of these crosses. It's there. Please make sure to practice the Punnett square, because that's really important from this particular chapter. In environment, the activities are very general, right? How you can reduce the waste production, how you will be behaving like an active citizen and like the fighter for our nature. So those activities are very self-explanatory. So we are not going to discuss about the activities of our environment. As I'm telling you, the activities are more of case-based. So in our environment, we have discussed there are only 3 important topics, right? Ozone layer, biological magnification, food chain, food web, and the 10% law of energy. These 3 topics are super important, so please make sure you go through them. So let's now everyone call Aishwarya ma'am, and we'll be ending the session now. So please make sure you go through these, all the important things we have mentioned. You don't have to worry about the diagrams, right? Just make sure to go through them, right? Please ma'am. This diagram is important, everyone, sex determination ka. And this we always say, that's how you have to practice. See, don't be scared. अब डरने से क्या होगा? Right? डरने से कुछ नहीं हो सकता है.
Yes, so everybody with this, if you see, right, end to end, we're done with biology, ma'am. We've done the Maha Marathon, the competency, important diagrams. We have done, you know, all the activities also, and we are done. So I hope all of you are feeling better now with biology. Your confidence level should be at least 100%, right, ma'am?
Yes, it should be super high, everyone. And can I tell you, there are still many of you who feel that you are scared. Now, there's a very small, a very thin line difference between having that a feeling of scariness, or basically not knowing things. So we are 100% sure that you are aware about the syllabus, you are aware about the concepts. You just have to have that fear of examination, right?
Yeah. And I think you should be more confident about what you have learned. Like, you should have confidence in yourself that yes, you have prepared, right? So very simple class, 1 hour mein fatafat we finished all 4 chapters. So as you all know, everybody, with this we come to the end, and of course after this you will be having your competency based questions with Saurabh sir. So everybody, make sure that all of you, right, please make sure that you go splash some water, drink a lot of water, and you know, eat some snacks as well, and get prepared to solve the questions. And all the very best. We'll be meeting you very soon on Friday.
Yes. Yeah, so please make sure, everyone, right, as I mentioned, have something. If you want to have your dinner, just be ready with the next class. And we'll say bye-bye. But before we go, please make sure you hit the like button for the video, share with your friends, and subscribe to this amazing channel, because we have so much more for you. We'll be coming very soon. We'll be meeting you again. Till that time, do take care of yourself, keep yourself healthy, and keep on learning with BYJU'S.
Yes, everybody. Bye. Take care.